Hey, Mr. P here. In this video we're going to talk specifically membrane structures. So we're going to talk about the anatomical structures, the components that make up the phospholipid bilayer, but we're also going to talk about how those components interact with each other. We're going to talk about how those components align themselves and anchor themselves within the membrane. We're going to talk about a little bit about membrane fluidity. We're also going to talk a little bit or get into the idea of membrane transport. That will be a future video by itself. But we're also going to talk at the end specifically about individual models that were and are used to help understand or help kind of drive our understanding of how these phospholipid bilayers or how these cell membranes align themselves and how they are structured. So in order to kind of understand the phospholipid bilayer or cell membrane, we first have to dive into individual components. It's no different than understanding anything else. If we can understand the foundational pieces that are put in place with a specific purpose in mind, we can have, in my opinion, a better full understanding of the structures as a whole. So we have to talk about phospholipid structure first. The phospholipid is a specific molecule that has a very descriptive or very specific set of characteristics. Those characteristics are fundamental and vital to the adaptations or the structural adaptations that have happened that have resulted in the overall structure and the success of that structure of the phospholipid bilayer. So what is a phospholipid? A phospholipid is a phosphate head, okay, and so you can see that there is a phosphate group within this head, and really the head is no more than a glycerol phosphate and what has kind of become a shortened fatty acid chain that has kind of come up and wrapped up not only the phosphate but the glycerol. So really, all of that aside, the only thing I think that you really need to kind of be aware of is that the phosphate head contains phosphate, right? It's called a phosphate head. It contains a phosphate group. This phosphate group gives the head portion of the phospholipid a polar designation. Okay, this is a polar molecule, or the molecule as a whole has a polar charge or a polar designation on the head portion of this molecule. The tails, in this case, are two fatty acid chains. Now, a phospholipid is a lipid. It's a special type of lipid. We talked about specific types of lipids in the lipid video when we talked specific structural adaptations of the lipid from a molecular standpoint. But fatty acids are just repeated CH2s. You can see CH2, 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 all the way to the end carbon, which is a CH3. We also, in that previous video, talked about the difference between saturated and unsaturated fatty acids. We also talked about polyunsaturated and monounsaturated. So if you need a review, go back to that video and review those. But these structures right here are very common or very familiar, or should be familiar to us at this point. Okay, we've already studied fatty acids. We know that fatty acids are lipid components. These fatty acids are no different than the fatty acids we talked about in that lipid video. But a phosphate head attached to two fatty acid tails constitutes this molecule that is known as a phospholipid. Now, this is kind of the in-depth chemical formula, so to speak, of a phospholipid. If we drop down into kind of this artistic rendition of a phospholipid, you will see that there is a polar phosphate head, and there are nonpolar fatty acid tails. Notice one head attached to two tails. When you matriculate or put these molecules into a structure of more than one phospholipids, you get kind of this unique bilayer effect. The reason for the phospholipids aligning in this bilayer is because there is water readily available outside. Let's just assume this is the outside portion of the cell, and there is water readily available on the inside portion of a cell. We know that we live in an aqueous solution. We have water in our environment. We know that we have a lot of water inside our cells, but there isn't, and I'm going to talk more about this in a little bit, there is no water, okay, no water on this inside portion because this inside portion is nonpolar. So by structuring our bilayer with heads out, you in effect create a zone of hydrophobicity which means hydrophobic zone. Again, I'm going to talk about hydrophilic and hydrophobic interactions on the next slide, but this is a zone of hydrophobicity, and so this region right through here has no water, while the heads keep this polar kind of boundary between all the water on the outside of a cell and all the water on the inside of the cell. So, as a recap, head, phosphate, polar, charged, and hydrophilic, which means attracted to water. These heads are attracted to water, so the heads are going to be attracted to water inside of the cell. They're also going to be attracted to water on the outside of the cell, aligning themselves in a bilayer with tails in, so that the tails, which are hydrophobic, which are repelled by water because they are nonpolar, remain kind of in this zone of hydrophobicity or this hydrophobic zone. So tails are made of fatty acid. There are 2 of them. They are uncharged, nonpolar, and hydrophobic. Okay, phospholipids are amphipathic. That brings up this kind of unique term, amphipathic. Amphipathic means that you are a molecule that has both polar and nonpolar regions. So both polar and nonpolar regions. Most molecules, like an individual phosphate, is just nonpolar. Okay, so it has no poles. It is nonpolar. It doesn't have any a various distribution of charges, right? It's kind of evenly distributed charge-wise around the whole thing. Water is a polar molecule. The whole molecule itself is polar. Phospholipids are unique because they have both polar and nonpolar regions. That makes them amphipathic. And so because they are amphipathic, they can orient themselves and always do orient themselves into this bilayer, which can be seen here. This lipid bilayer, when we kind of expand it out with more and more phospholipids, you start to see that it does really create this sheet effect. And because, again, there is water readily available outside of a cell and water relatively or readily available inside a cell, it orients itself into this sheet that keeps all of this interior region free or completely void of water. So no water, while we do have water inside and outside. So hydrophilic are the heads, hydrophobic are the tails. Again, no water on the inside. Depending on the number of phospholipids, they can create a couple different structures within water. Let's just assume we dumped a whole bunch of phospholipids. If we had individual phospholipids and we dumped a whole bunch of them into water, they would naturally form these structures. Now, if we had a bunch of individual lipids, they likely would produce something like this. Now, this liposome is cut in half, so we're looking at the cross-section, but you will see that there are heads on the inside to kind of create this individual void or cavity, and there are phospholipid heads on the outside. So this begins to take shape or begins to look like an individual cell membrane, right? And that individual cell membrane begins to provide compartmentalization that eukaryotic cells and prokaryotic cells really need in order to thrive. When we look at this kind of individual liposome structure, you start to see that it really is just a folded up bilayer sheet. But in smaller quantities, these phospholipids can actually just create these micelles, and that's essentially just a kind of a lipid, kind of like a lipid bubble, so to speak, right? So if we dropped vegetable oil or olive oil into water, the little tiny kind of droplets of oil would create these micelles, and those are available and kind of free-flowing within the cell cytoplasm also. It just kind of depends on the number of molecules, but the main important kind of the concept that really needs to be addressed by this is that while it isn't necessarily vital that you understand what a liposome is or what a micelle is, you do understand that the heads are going to orient themselves out towards water, and the tails are going to orient themselves in, and that the phospholipids are going to create a bilayer. Fatty acid tails do not attract each other very strongly, leading to the fluidity of the membrane. That's really important as well. It's called a fluid mosaic model that I'm going to be referring to in a little bit. It is fluid. This bilayer does move, okay? So if you can imagine this mat floating on top of water where it kind of undulates up and down with the flow of water, with the waves, that's very indicative of the way the bilayer actually looks. phospholipids can flow or move past each other laterally, but they can't move vertically. What that means is that if I highlight this phospholipid polar phosphate head and then allow this membrane to kind of interact with each other over the period of a few minutes, I would likely see this membrane phospholipid move and is now located here. And then in the next 2 minutes, maybe it's back here. In the next 2 minutes, maybe it's up here. So all these membrane phospholipid molecules are constantly moving past each other laterally, but you will not see this particular head flip-flop and come down here. Okay, that lateral movement cannot happen, but the side-to-side kind of, or I should say this vertical alignment, vertical movement can't happen, but the lateral movement side-to-side can. This allows animal cells to have a variable shape and also allows the process of endocytosis to take place. Endocytosis is the form of cell eating. It is a form of bulk transport. Again, we're going to have another video specific to membrane transport that will talk in depth about active transport and bulk transport. But for now, just know that endocytosis does happen, and the reason it happens is because animal cells in particular have the ability to remain fluid and also have the ability to kind of move these materials or these components, those phospholipids, in a kind of lateral way. Moving out of the phospholipid, because I think we get that the phospholipids are oriented in a bilayer, we have to talk about the protein components of the membrane, because while the phospholipid is incredibly important, the proteins really do the stuff for the cell. Phospholipids create the membrane, so to speak. The proteins do the functions that the membrane needs to do. So, proteins that are embedded in the membrane are extremely diverse in their structuring, but they're also extremely diverse in their functioning as well. Proteins are embedded in the fluid matrix of the phospholipid membrane or the phospholipid bilayer, and the proteins really in concert with the bulk of the membrane, which is the phospholipids, really do begin to start creating that mosaic effect. When you look at a mosaic in art, or when you look at a stained glass window, for instance, which we often refer to as a mosaic, you will see individual components that when by themselves don't really create the masterpiece, but when put together into the orientations that they're put together, it really does create kind of this really beautiful work of art. And that's the way the cell membrane works. All of these individual components are put together in concert with each other, kind of spread out throughout the membrane to really work perfectly in concert with each other, and it helps to create this fluid mosaic model that I will again explain in a little bit. There are two main types of proteins I want you to understand: integral proteins and peripheral proteins. Peripheral proteins are just like what it sounds like, the periphery. So your peripheral vision is the vision you have on the sides of your head. So those are the kind of extreme outside vision field. So when you refer to peripheral proteins or proteins as peripheral, these are the proteins that do not protrude through or into the middle of the membrane. So peripheral proteins, for instance, are these structures here. Now I know in this particular diagram, these proteins do semi-protrude into the middle, which kind of contradicts what I wrote here. However, for the most part, peripheral proteins don't expand into this middle nonpolar region of the membrane very far, and or don't span the entire bilayer. When you notice or contradict, kind of compare these peripheral proteins with these proteins, for instance, you will see that they kind of stick out of this side of the membrane and protrude out that side of the membrane. So that is an integral protein. So integral proteins span the entire membrane. They span the entire membrane. They are permanently embedded, and they are polytopic, which means that they are multi-surface proteins, which means, just like I said, they extend past this surface, so they are interacting on this interior surface, and they are extending out this surface, so they are interacting on this exterior surface as well. These are your integral proteins. This one is, 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 this one is. These that are not multi-surface or interacting with multiple surfaces, these are your peripheral proteins. And because they are peripheral proteins or structured on the periphery of the membrane, they do not protrude into the middle hydrophobic region, They remain bound to the surface of the membrane, and again, that can be exterior surface or interior surface. Notice this is an interior surface protein. This is an exterior surface protein. They usually have a temporary association with the membrane, which means they, like the phospholipids, can move. Okay, so this protein can move around laterally, just like these phospholipids can move around laterally. The reason that these integral proteins are permanently fixed is because they are literally anchored in both polar surface regions, but also anchored within the nonpolar region as well. And because they have so many kind of anchor points, they are essentially permanently fixed. These peripheral proteins, because they are temporarily associated with one surface only, they are considered monotopic, which means they are one surface oriented. Glycoproteins are exactly what they sound like. Glyco is short for sugar, and protein is obviously protein. And so these are specific proteins that are kind of anchor points for this oligosaccharide short chain of carbohydrates, right? So an oligosaccharide, just like monosaccharide, disaccharide, polysaccharide, saccharide is sugar. Oligo just kind of means short. So these are the short chains that you see here. Each one of these is an individual monosaccharide. When we embed or link all of these monosaccharides together, you get a polysaccharide. But in this case, it's a really short chain. And the purpose of these glycoproteins is all about surface marker. These are essentially the name tags. These are the structural components that are kind of communicating between cells, which is incredibly important. Cells have to communicate with each other. These little red guys that are embedded within this hydrophobic region are the cholesterol molecules, and cholesterol controls membrane fluidity, which also controls rigidity. So these are essentially the mortar that is holding the bricks, which are the phospholipids, together. The more cholesterol you have, the more mortar you have, the more structurally sound your wall is. The less mortar you have, meaning you just have kind of bricks dry stacked, the less rigid that layer is. When we talk about cholesterol a little more, cholesterol has a hydroxyl group, which makes one end of the hydrophilic region, or one end polar and hydrophilic. This is the hydrophilic region. It is the region that anchors that cholesterol molecule to the actual polar head region of the phospholipids. They also have 4 carbohydrate rings. That is the steroid component. And then they have a nonpolar hydrophobic tail, which is how the cholesterol molecule down here anchors itself to the fatty acid specifically. So you have kind of a polar region that is going to attract itself to the head. You have a nonpolar region that is going to attach itself to the tails. And then you have the actual steroid that helps to kind of do the things that the cholesterol molecule is supposed to do. Now let's talk specifically about membrane protein structure and function. You will see that there are a variety of membrane proteins, and each of them look different because each of them act different or have a specific purpose. Some of the protein functions, and again, these are fundamental to the success of the membrane, and things that you absolutely have to know is that several of the proteins have receptor purposes. So this is a receptor protein. A receptor protein has a specific job to receive or dock chemical messengers. Those chemical messengers can come from enzymes, for instance, okay, or they could come from an adjacent cell. Maybe this is an adjacent cell. You kind of can see this phospholipid bilayer section. We know that there are junctions between cells. If this is one cell or cell number 1, and this down here is cell number 2, these receptor molecules have to receive signals that are sent from other cells. Okay, so these are sites for hormone binding. If one cell releases a hormone, another cell has to receive that in order to produce an effect within that cell. And so the way that these receiver cells, the receptor cells, work is that when the hormone is received, it causes a conformational change or a conformational shape change to this particular protein, which results in a message being relayed into the interior of the cell. Another function of membrane proteins is enzymatic activity. So there are enzymes that are embedded in the membrane that have the purpose of enzymatic action. Again, we've had previous videos on enzymes and enzyme action. And so these enzymes break down things, right? So these chemical messengers could be broken down into byproducts, these chemical substrates of various concentration or various type can be broken down into products. A lot of times the cell needs certain molecules broken down, and they employ their enzymes to do that. Then we have channel proteins. Now those channel proteins, like these two, look or can look similar, but have vastly different functions. Channel proteins, true channel proteins, are utilized within a passive transport context. So this would be a passive transport channel. Notice it is open from outside to inside, and the purpose of passive transport is to move solutes from an area of high concentration to an area of low, and they will just, those solute molecules will naturally diffuse down a concentration gradient without energy, without the need to guard these cells, without the need to use or spend ATP. However, when we want to pump these solute molecules from low to high, we physically have to input energy, open these gated or voltage, sometimes voltage gated channels, but a lot of times use energy to physically move these proteins so that these molecules can be pumped against their concentration gradient from inside to out or outside to in, but from a low concentration to a high concentration. We already talked a little bit about glycoproteins. Glycoproteins are those proteins that are associated with an oligosaccharide. They are all about cell surface markers, cell communication. Okay, so those carbohydrate chains serve as identification markers and communication between the cells. And then these CAM, cell adhesion molecules, are all about attachment sites between adjacent cells. So this cell can bind itself to this cell to hold these cells together through this kind of junction, and it's all about cell adhesion so that those cells can structure themselves into more complex tissues, which can kind of increase the functioning of those cells. Kind of the whole concept is kind of that strength by numbers idea. So now that we've talked a lot about kind of the purpose of the individual components through phospholipids, cholesterol, and then all of the various proteins that come together to produce the fluid mosaic, now we can kind of step back and look at this structure in a more holistic way. We know that we have these proteins that span the entire membrane. We know that we have these kind of peripheral proteins. We know that we have these individual cholesterol molecules that are linking, kind of adding structural integrity to the membrane. And then, of course, we know that we have all of these phospholipids that are orienting themselves into this bilayer because they have to always orient themselves heads towards water. And because we have water in this extracellular fluid and we have water that makes up the cytoplasm, we know that the tails have to be interior of the cell phospholipid bilayer and heads have to be exterior. But the membrane does remain fluid, and you kind of can tell within the undulation of this particular diagram. And so it is important to regulate the degree of fluidity, and the way that it does that is through the cholesterol. So fluidity is regulated by cholesterol. You have to be fluid enough to allow cell movement. Okay, the cell has to physically be able to move. We have to also be able to move the required substances, but if the membrane is too fluid, you can't adequately regulate the movement in a controllable way, and so you can't restrict movement of substances across the membrane. Things would just be able to push their way in. The cell wouldn't be able to hold a very structurally supportive shape. And so the way that cholesterol regulates this is that cholesterol restricts the movement of phospholipids and other molecules. The way that I like to think about this is whether you think about it in terms of the mortar between bricks, which kind of helps set the bricks in a more fixed, rigid way, or if you like to think that this is kind of the adhesive tape or maybe the Velcro that holds these molecules together. It just increases friction, and because we're increasing friction, we're decreasing movement, therefore restricting movement, therefore causing rigidity and structural integrity to the membrane. So the Singer-Nicolson fluid mosaic model is all about putting all of these individual components into this entire structure in a very prescriptive way. Phospholipid molecules form a bilayer. We've talked about that. Phospholipids are fluid and move laterally, not vertically. Peripheral proteins are bound to either the inner or outer surface of the membrane. Integral proteins span the entire thickness of the membrane. The membrane is a fluid mosaic of phospholipids and proteins, and what I mean by fluid mosaic is that it is a mosaic because it is a large component made up of individual smaller components, but it's also fluid because it has the ability to move and undulate and move the individual components laterally so they are not fixed or rigid within the structure that is the membrane. Proteins can move laterally along the membranes along or as long as they are not kind of permanently fixed, like a lot of the integral proteins are. Some of the evidence for this model or some of the things that have come about to strengthen this model is that membrane proteins were found to be varied in size and not uniform and globular in shape, which means, as you can see here, that there isn't a fixed shape of proteins. Some of these proteins are very long, thin, and drawn out. Some of them are long and span the entire membrane. Some of them are short and globular. Some of them are primary or secondary conformation. Some of them are more tertiary and quaternary conformation. The evidence is pretty straightforward, right? And I know I haven't talked about this alternative models that help to disprove or help to prove the Singer-Nicolson model, but I will in a little bit. These globular protein shapes would be unable to form continuous layers. This is the important part of this fluid mosaic model. Because of the varied size and the varied structure and the varied process kind of purposes of these membrane proteins, it would be unable or would be impossible to form a continuous layer on the periphery of the membrane, which is really kind of the only evidence that the original model had based on pictures. And again, I'll show you a picture in a minute. These proteins had hydrophobic and hydrophilic regions, okay? And the use of fluorescent antibody tagging resulted in our understanding of membrane protein movement. Red and green tagged proteins after several minutes were evenly distributed throughout the membrane. Basically, it allowed us to understand this fluid, kind of this fluid motion of the phospholipids. We were able to radioactively tag some of these phospholipids with green and with red, and they didn't stay the way we tagged them, right? If we tagged all of these over here with red and we tagged all of these with green, after several minutes, basically the green and the red were all evenly distributed, which basically leads us to conclude one thing, and that is that these phospholipids can move laterally, and therefore they do. So all of this evidence led to the proposition of this new model and helped to discredit or disprove the previously accepted model, which is this Davson-Danielli model, which I'll talk about in a minute. So what is the Davson-Danielli model? This is a model that was previously accepted up until the 1950s when the Singer-Nicolson model, well, I should say proposed in 1972. Okay, up until the 70s, this Davson-Danielli model was the model that was accepted within the scientific community. And basically, based on old images, okay, the images at the time, when you looked at this phospholipid bilayer, it looked to be two very thick structures with a very thin middle. And so the way that they concluded that this structure should have or could have come about is with this illustration. They said that the phospholipids that make up the membrane are sandwiched between two layers of very uniform proteins. Okay, so in a sense, there is a protein sandwich that sandwiches or completely encases the phospholipid bilayer. How is that wrong, or how was it falsified? Basically through the technique called freeze fracturing. So the way that this works is that you rapidly freeze cellular components. In this case, we specifically froze using liquid nitrogen the cell membrane, and then through the fracturing process was actually able to create the fracture between the bilayer. And so we essentially opened up this phospholipid bilayer like a zipper, which you can see here. We basically took this part off and we kind of opened it like a Ziploc bag, so to speak, and found that the inside lipid bilayer, when viewed under microscopy, actually appeared to have all of these really rough indentations or rough kind of bulging structures, which was very different to the way the Davson-Danielli model said the membrane was structured. And so upon closer inspection of what these actual nodules are is that the irregular rough surface was actually interpreted to be integral proteins that were spanning through the membrane. So in effect, we took this layer up and this layer down, and because there were these individual bumps, these were in fact the proteins that were spanning through the membrane that now became visible. And because we could now tell or see that there was in fact membrane proteins that were spanning the surface, it contradicted the way the Davson-Danielli model was articulated at the time. And that was mostly because the model didn't include any transmembrane proteins through the structural kind of middle of the membrane. It just talked about that kind of uniform protein sandwich or protein coat that was on the outside. So because we employed freeze fracturing, because we saw these images, we later learned that there are in fact irregularly sized proteins, and those proteins span the entire membrane. That's the end of the lecture. Kept it to 30 minutes. See you in class. Bye.